We online too? Okay. So it is my distinct pleasure to introduce Dr. Banrida Wahlang. She is currently an assistant professor in the Department of Medicine, Division of Hepatology, Gastroenterology, and Nutrition. Her research background focuses on studying impacts of environmental exposures, including persistent and volatile organic pollutants on liver and cardiometabolic diseases. Dr. Wahlang received her Ph.D. from the University of Louisville in this department, and I'll throw in a personal note here, which is that I met Banrida in New Delhi when she guided me around the city. I was visiting, and it was a result of that lucky encounter that brought her to Louisville.
She always does that in this conversation.
Well, I promoted the programs. You came with your own initiative. Anyway, okay, so she completed postdoctoral training in the UK in the Superfund Research Center for her first postdoc, and second was here at the Superfund Research Center. So she's a Superfund trainee through and through. Her current work focuses on sex differences in the environmental health sciences and underlying sex-dependent mechanisms that drive disease outcomes with chemical exposures. She received an NIEHS K01 award to help fund this research, as well as a CIEHS Pilot Award. And her research interests and goals also include studies that broaden from women's environmental health to sex-dependent transgenerational effects, as well as developing intervention strategies to combat pollution effects on humanity. We're very pleased to hear from her. Banrida, the floor is yours.
Thank you. Okay, I hope the microphone is working. Okay, so thank you for that lovely introduction. I remember being invited to give this kind of talk about 4 years ago when CIEHS was still putting itself together, and I think I was one of the trial runs for these EHS seminar series. I'm glad to be invited back, and it's my pleasure to be here. So today we're going to talk about mechanisms of environmental contributions to fatty liver disease. And I'm going to start off by talking about what is fatty liver disease. So as the name implies, fatty liver disease basically means a deposition of excess amount of fat in the liver. So this is clinically also called as hepatic steatosis. And this is important because the liver is a really essential organ with many life-supporting functions. So we want it to be always healthy and functioning. So in broad categories, fatty liver disease can be split into two parts based on the cause. So the first one, which is very well known, is alcohol-associated liver disease, or ALD. It has about a 0.5% global prevalence, and it's mostly prevalent in females, and it's caused, as the name implies, by heavy alcohol consumption. The second category of fatty liver disease that is non-alcohol based, it's called non-alcoholic fatty liver disease. It has a higher global prevalence of about 25%, and it's mostly seen in males as well as postmenopausal females. So in terms of pathogenesis and liver pathology of this disease, it somehow is the same for both these categories of liver diseases. So it's irrespective of the cause, the disease progression is very similar. So it can start by having a normal liver. And if you can look here, this is just a histology of how a normal liver section looks like. And fat accumulation in the liver results in what is called steatosis. And you can see these white little droplets here. And this could progress further and become inflamed and have neutrophil infiltration and cytokine proliferation to cause steatohepatitis, which is basically a fat plus inflamed liver. And it could proceed to fibrosis and tissue scarring, which is cirrhosis, and also progress to hepatocellular carcinoma or liver cancer. And this could inevitably result in fulminant liver failure, and it could be resolved by either a liver transplant or could cause or result in a liver-related death. And so if you look at the percent of populations, for instance, for ALD, it seems to be more aggressive because about 90% of heavy drinkers tend to have steatosis, and about 20 to 40% of those tend to develop steatohepatitis, and about 8 to 20% for cirrhosis, and 3 to 10% for liver cancer. And for NAFLD, it is somewhat less aggressive compared to ALD, where you still have about 25 to 30% of the population developing steatosis, and about 70-30% developing steatohepatitis, about 44 to 14% for cirrhosis, and about only 0.2 to 0.5% for hepatocellular carcinoma or liver cancer. So there are a number of factors that could contribute to non-alcoholic fatty liver disease, and it could also be caused without any pre-existing conditions. But these number of factors that have been identified over the years include being either overweight or obese, having symptoms of the metabolic syndrome, which could be high blood pressure or visceral obesity or high triglyceride levels in the blood, having type 2 diabetes or insulin resistance. Also having obstructive sleep apnea could also lead to a fatty liver disease due to intermittent hypoxia as well as metabolic disturbances. Asian or Hispanic individuals are more at risk for fatty liver disease, as well as postmenopausal females or females having polycystic ovarian syndrome. But the most recently identified, if you will, or more recently discussed risk factor for fatty liver disease has been exposure to environmental chemicals. And the reason why the liver is so susceptible to environmental chemical exposure is because it's the common target organ for chemical toxicity. So it's basically trying to do its job to get rid of the chemical, and it's getting attacked by the chemical. And this graph over here just show a window in one of the studies a review article that had looked at this EPA IRIS database that showed the percent of chemicals and what kind of target organs did they impact. And you can see here that the liver is the most impacted target organ for chemicals. And it's also important to remember that we live in a very globalized world. And so even in 2022, which is this year, about 194 million chemicals were registered with the American Chemical Society. And that is a huge number of chemicals. And hepatotoxicity or liver toxicity has been documented for about 1/3 of about 600 chemicals that are in the National Institute of Occupational Safety and Health Pocket Guide. So in terms of environmental contributions to fatty liver disease, I was trying to see how many publications are there that have, kind of addressed this topic. So there are about over 3,000 publications documented just in PubMed alone that had looked at environmental chemical exposure and liver disease. And these include non-alcoholic fatty liver disease, and a lot of the chemicals associated with non-alcoholic fatty liver disease include persistent organic pollutants like dioxins, volatile organic compounds, while other kinds of liver injuries that were also documented include alcoholic steatohepatitis. So there was this one study that looked at this northern contaminant mixture, which is basically a mixture of persistent organic pollutants, heavy metals, and flame retardants that is widely seen in the Inuit population in the Arctic region. And they looked at these chemical exposures along with alcohol consumption and found that this particular northern contaminant mixture actually exacerbated alcoholic steatohepatitis. And other kinds of liver injuries, including hepatitis B and hepatitis C viruses, as well as primary biliary cholangitis, which is basically injuries to bile ducts, have also been seen with chemical exposure, as well as liver cancer. So for the most part, these chemicals induce or mediate these toxic effects on the liver through a number of ways. It could be steatosis or oxidative stress or inflammation or cell death or apoptosis. And so this huge array of liver injury that these environmental contaminants have impacted led us to do some more research on it as a group, and we came up with this. Actually, Matt Cave came up with this word called toxicant-associated fatty liver disease and toxicant-associated steatohepatitis, or TASH. So if you meet him, he'll say, "Hi, I'm Matt Cave," and I coined the word TASH. So in terms of TASH and TAPLD, this is widely seen in chemical workers initially, and there's still limited data on sex differences. So there's no consensus to say that, females are more prone to or TASH is more prevalent in males. But one important thing about TAPLD and TASH is that it depends on the chemical that you're exposed to, for how long you're exposed to, and how much you're exposed to. And so just for a historical perspective, looking back on how these evidence of environmental chemicals have, affected the liver, this dates back all the way to the 1960s and 1970s when there were these accidental poisoning that happened around the world. For instance, there was this well-known Yuchen disease that happened in Taiwan where thousands of people were contaminated with polychlorinated biphenyls or PCBs when they ingested this rice oil that was contaminated during the manufacturing process. And closer to home in Louisville, Kentucky, in the 1970s, there was a physician from a BF Courage plant that had identified 4 chemical workers that were working in a polyvinyl production plant to be having this rare form of cancer called hepatic hemangiosarcoma, which is cancer of the blood vessels in the liver. And so all these things were happening. And in 2010, that was when this word TAFLD and TASH was coined by our group. And during this year, there was this study that came out that I think kind of revolutionized the TASH and TAFLD world, in which there was these evidence of steatohepatitis that happened that was seen in these polyvinyl chloride production workers. And so 10 years later, in 2019, there have been some clinical practice guidelines that have been issued, at least in terms of occupational liver disease. And I think this was with the European Association for the Study of Liver Disease. And I believe, hopefully, we will have some of those guidelines as well in the States. So let's just talk a little bit first about this first class of chemical called volatile organic compounds and how they are related to liver disease. So VOCs are very volatile. That's why they're called VOCs, and they have a very high vapor pressure, so they tend to evaporate easily into the atmospheric air. And they're widely found in a number of household products, candles, scented candles, cosmetics, even during cooking processes. And so they're a major issue in terms of indoor and outdoor residential air exposures. And a number of these VOCs have been found to be presented at a number of Superfund sites. So even though they're present in the air, they could also be found in the groundwater and seep through contaminated sites into other residential areas in proximity to those sites. And exposure to a variety of these VOCs have been correlated with steatosis, which is again fatty liver, liver injury, etc. So a very widely studied VOC is vinyl chloride, and like I mentioned, it has been associated with liver cancer. But we were talking about occupational exposures that were really high dose, high level exposures. But what happened when we're talking about exposures that we're currently used to today, these residential exposures? So a more recent study that was done within the decade by one of our ex-faculty here in PharmTox, Dr. Eula Bayer, they did a study where they looked at vinyl chloride exposures at doses that were way below OSHA limits, which is around 1 parts per million. And they found that those low doses of vinyl chloride, when mice are exposed to it, they do develop metabolic disturbances in the liver, as well as it also enhanced diet-induced liver injury. So if you add a high-fat diet and expose these mice to this low-dose vinyl chloride, it also exacerbates TASH. And there are also other studies that have looked at other types of VOCs rather than vinyl chloride. So our group have looked at blood, benzene, toluene, xylene, and styrene. These are compounds that are associated with the Deepwater Horizon oil spill. And we found that these blood BTEX levels and heavy metals are also associated with liver injury in Gulf state residents. And benzene exposures have also been shown to induce metabolic dysregulation, including insulin resistance in mouse models, as well as dysregulated glucose metabolism. So in a recent study that we performed as part of the Superfund Research Center Project 1, this was a study that was done in participants or community residents of Louisville from 6 neighborhoods in South Louisville. And this study is also called as the Health, Environment, and Action in Louisville, or the HEAL study. It was done in partnership with Greenheart. And sample collection for this study happened way before COVID-19, so between 2018 and 2019. And so the major finding from the study was we were interested to know if these ambient VOCs or residential VOC exposures could have any effect at all on liver disease biomarkers. So these participants, after giving the informed consent, we would collect urine, blood, etc. from them. And so we were able to measure about 16 urinary VOC metabolites from these participants, as well as 7 liver disease biomarkers, which included liver enzymes, cell death markers, and direct bilirubin. And the major finding from the study was that we saw that about 8 VOC metabolites, which corresponded to parent compounds, namely acrolein, which is widely found in cigarette smoke, acrylonitrile, crotonaldehyde, styrene, and xylene, were positively associated with elevated ALP levels. So ALP is an enzyme that in the liver that when it's elevated, it is usually indicative of some sort of injury to the bile ducts, etc. So it's associated with to an extent with cholangitis and primary biliary cholangitis. And so another VOC that was also associated with a liver disease biomarker was benzene, and it was associated with serum bilirubin. And we found that these associations were more pronounced in smokers, and it's just sort of reiterate that, smoking may be a risk factor for liver injury and dysfunction. And that was, and these doses were residential low dose exposures. Some of the currently known mechanisms for VOCs based on all those tox studies and epi studies that were done was primarily VOCs are thought to act by forming binding to proteins to form adducts because they do get metabolized into reactive intermediates. And so they induce this carbonyl stress, if you will, on cell organelles, including mitochondria, leading to oxidative stress, lipid peroxidation, as well as induce endoplasmic reticulum stress. VOCs also act as metabolism disrupting chemicals, so they disrupt normal hepatic energy metabolism by interfering with kinase function. So for instance, vinyl chloride at low doses has been found to activate both the AMP protein kinase and mammalian target rapamycin or mTOR, and both of these are energy sensors that are usually activated in opposition of each other. But vinyl chloride paradoxically activated both arms simultaneously, leading to a metabolic disarray. And other mechanisms with VOC exposures include cell death activation. And again, this liver cell death caused by these VOCs is also kind of a very a tricky mechanism, if you will, because with low dose exposures, it's usually through apoptosis, and in occupational workers, we've seen cell death markers that are mostly related to necrosis that are elevated. So we're going to switch a little bit to another group of chemicals called persistent organic pollutants. This is my favorite class. I've worked with these chemicals for years. And they're not my favorite chemicals, but they're just a great chemical to do research on. So persistent organic pollutants, as the name implies, persist in the environment. They're resistant to degradation, and they're also known as forever chemicals. Unfortunately, POPs were man-made. A lot of them were produced intentionally for industrial processes, while others were produced as byproducts or inadvertently. And so in 2004, there were a number of countries that came together in this POPs convention in Stockholm that decided to ban or regulate 12 chemicals or 12 persistent organic pollutants, also known as the Dirty Dozen. It included a number of organochlorine pesticides, Pcbs, dioxins, and then dibenzofurans. And since this convention, other POPs have also been added, including the PFOSs, PFOS, as well as other pesticides. And I think this list keeps getting bigger and bigger as we move through the years. And so one question that I get asked a lot in POPs research is, okay, so we have produced these chemicals 30 years ago. They've been banned by US Congress in the 1970s. Why do we even care to study about them still? So one of the reasons why we still want to study about them, A, obviously they're forever chemicals, but B, there are other countries that are, a lot of other developing countries that are still, not regulating these POPs production and use as tightly as Europe and North America. And so POPs have the ability to migrate throughout the globe through this process called grass hopping, where they tend to evaporate at lower altitudes and deposit themselves at mid and high latitudes, and it just keeps going on and on. And so even though we're seeing that some of these serum POPs levels have gone down in human samples and in the States, it's still a major concern because of this, this likelihood that they're still migrating and they can show up still. And another thing with POPs is they're very hydrophobic, they're resistant to metabolism, and they could bioaccumulate in living organisms. So they biomagnify to the food chain, and we are basically carrying them and passing them along, if you will. So that's why we still study them. So in terms of POPs and liver disease, one of the POPs chemicals that have been widely studied is Pcbs and dioxin. So a number of studies have shown correlations between Pcb and dioxin exposure with both TASH as well as fatty liver. And in terms of Pcbs, this is the structure over here. As you can see, it's a biphenyl, and it could have chlorine substitutions at any of these rings. So depending on those chlorine substitution, you can have up to about 209 congeners. And these Pcbs were initially manufactured by this chemical company that you all may have heard of called Monsanto Corporation, based in Anniston, Alabama. That was where they are headquartered, their manufacturing plant. And they used to produce these Pcb congeners as mixtures and sold them under this brand name called Aerochlor. So you'll hear me talk about this Aerochlor a lot, which is basically a mixture of Pcbs. And our group and others have shown that there are positive associations between circulating or serum Pcb levels, as well as organochlorine pesticide levels, as well as PFASs, with liver disease biomarkers. And these are just some of the studies that were done in the National Health and Nutrition Examination Survey NHANES population that is very reflective of the American general population. This particular study here looking at PFAS exposure and liver disease biomarkers, I think is one of the very first studies that have been done to kind of establish this relationship between PFAS and TASH and fatty liver disease. And this study was done on the CIEHS population from West Virginia. So many of you have probably seen the movie Dark Waters, and that's where that population came from. So, okay, we are a little stuck. What about toxicology studies on PCBs and liver disease? So we have mouse model systems that we've done through the years. I think we started doing our Pcb exposure studies in mice about 10 years ago. And so in our first study, we had selected this particular Pcb congener called Pcb 153. And the reason we used it was because it was present at really high levels in the highest levels in the NHANES population. But what was really important, a finding from the study, was that we found that Pcb 153 is a diet-dependent obesogen. So obesogen are chemicals that help promote obesity, and so Pcb 153, in conjunction with a high-fat diet, could exacerbate that visceral adiposity as well as fatty liver. And some of the mechanisms that we thought at the time that played a role include increase in lipid biosynthesis as well as decrease in fatty acid oxidation in the liver. And so since then, we've performed a number of other studies looking at not just 153, but also different kinds of congeners, including the Aerochlor 1260, which is a Pcb mixture, as well as the dioxin-like Pcb 126. And in most of those studies, we've done where we look at not just chemical exposure alone, but also in conjunction with either a high-fat diet or a diet that could induce steatohepatitis. So we had this concept of a two-hit hypothesis where the first hit could be, a high-fat diet, and the Pcbs were acting as a second hit to exacerbate the diet-induced steatosis. And in part, we thought that those mechanisms for those exacerbated steatohepatitis could be through activation of xenobiotic receptors, such as the aryl hydrocarbon receptor, AHR, the CAR, and the PXR. And based on those tox studies that have been done, so some of the currently known mechanisms for POPs, especially for Pcbs and dioxins and an extent PFASs, has been through this hepatic receptor activation. So POPs are xenobiotic receptors, so naturally they go and activate the xenobiotic receptors to induce their own metabolism, but because they're such hydrophobic and big molecules, they take a long time to get metabolized. So they have half-lives that are about, some of them have about 15 to 20 years. And so they could activate these xenobiotic receptors, but it could also activate other receptors in the liver that are involved in endobiotic processes like lipid metabolism, bile acid metabolism, etc. So this picture here just kind of show how POPs could activate these hepatic receptors and what are the downstream effects that happen from activation of those receptors. So for instance, activation of PXR, CAR, and AHR induce the drug metabolizing enzymes like CYP450s, but also induce transporter expression. And so for some of the POPs that rely on these transporters to get into an organ, this becomes even more relevant. Another known mechanism for POPs is hepatic signaling disruption. So PCBs have been shown to inhibit this epidermal growth factor receptor, or EGFR, and also inhibit the AMPK signaling cascade. And this inhibition of kinase activity eventually could also result in changes in metabolic gene transcription and eventually result in metabolic disturbances. And another known mechanism for POPs is endocrine disruption. So POPs have been shown to interfere with normal hormone function, including hormones like leptin and insulin, which eventually could result in metabolic disturbances. And so this ability of POPs to both disrupt, hepatic metabolism as well as the endocrine system led to this hypothesis called as the metabolism and endocrine disrupting chemical hypothesis that was probably proposed about, I think, 4 or 5 years ago. So this is important because when we're talking about endocrine disrupting chemicals, a lot of them, especially organic chlorine pesticides, have mostly been associated with diseases that are associated with the reproductive endpoint. So, when we're talking endocrine disruption, we're thinking breast cancer, ovarian cancer, PCOS, prostate cancer, etc. But more recently, studies have also shown the ability of these chemicals to promote metabolic changes that could result in obesity, diabetes, or fatty liver. And this is facilitated because of the role of the endocrine system in maintenance of energy homeostasis. So insulin, leptin, and sex hormones do play an important role in regulation of lipid metabolism and carbohydrate metabolism, etc. One of the principles of the EDC-MDC hypothesis is that it has to be a receptor-based mode of action because they do have to activate these receptors that play a role in both endocrine function and metabolic function. They also could be non-monotonic dose response, and the reason for that is because hormones themselves have a very non-monotonic dose response. They may require a second hit, for example, a high-caloric diet or alcohol consumption or another chemical exposure to cause those disruption. And some of the effects that you see with these chemicals are very dissociated from what we normally would think could be, components of metabolic disruption. So you could see a fatty liver but no obesity, or you could see fatty liver with no insulin resistance. Another important concept to remember when we're talking about this EDC and MDC hypothesis is the duration and timing of exposure. And this leads to what we also may discuss later, which is the developmental origins of health and disease. So there was this great study that was done recently where they look at a PFAS contamination in water and looking at in utero exposures and how that could alter hepatic lipid metabolism, etc., in children and eventually result in childhood liver disease risk. And another thing about these EDCs and MDCs are also they could have sexually dimorphic effects leading to sex differences. So some of the other emerging new concepts for POPs. So this hepatic receptor activation was not sufficient to explain some of the other transcriptional reprogramming that was seen in the liver. And so epigenetic mechanisms have already been established for POPs toxicity in terms of cardiovascular disease and cancer endpoints. And so more recently, changes in DNA methylation and circulating microRNAs with Pcb exposures were performed for this heavily exposed population from Anniston, Alabama, known as the Anniston Community Health Survey, or ACHS. And so our group had done this particular study on the ACHS population that was just recently published in EHP, where we look at circulating microRNA levels, and we found that there were these Pcb-mediated associated microRNAs that were changed that were also related to liver disease, including microRNA 122 and 192, and we were able to validate those mRNAs expression in an animal model as well. Another new concept of mechanism for POPs-mediated toxicity in the liver is liver epitranscriptomics. So we talk about DNA epigenetics and DNA methylation, right? So the RNA could also be modified post-transcription, and this is known as RNA epigenetics or epitranscriptomics. And our group have also looked at this particular mechanism, and this was done under the leadership of Dr. Carolyn Kling. So we had given them these livers of these animals that were exposed to different types of PCBs, and they did a global chemical RNA modification identification using LCMS, and found that PCB exposure caused significant changes in this global hepatic transcriptome in the liver, especially for M6A, which is basically a methylation that occurs at the nitrogen position 6 on the adenosine base. And this is also associated with altered expression of enzymes related to the RNA modification that are also known as readers and writers. And there's more work coming from this group, and I'm not going to talk about it because I'm sure Carrie will be able to update this group about that in the future. Some of the emerging new concepts that I'm personally interested in is sex differences. So the reason why this is so important even now, so 4 years ago when I said I wanted to study sex differences, it was great. So it's been 4 years now, and people are like, okay, everybody is doing it. But I was talking with another person from another university that also does this kind of research, and she was telling me that people are picking females and adding them to the males, which is great, but nobody's really doing it right. Like, you're not going from the basics. So that is something that I want to do, and I hope that some of the findings that I'm presenting here could show that as well. So sex differences is important because till like a decade ago, there was this general belief that men and women did not differ significantly in response to drug treatment, if you will, and there was this lack of inclusion of women in clinical trials and other health studies, just because it could introduce additional variables like pregnancy and lactation and use of oral contraceptives. So in 1985, the PHS Task Force on Women's Health concluded that healthcare has been compromised for women due to this lack of research. And so the US FDA and the NIH now require all biomedical research to address sex, gender, as well as racial and ethnic representation in clinical studies, and as well as in research studies to an extent. And so sex and gender is really important in EHS research or environmental health research because they do dictate the amount of exposure we get to chemicals. For instance, as an example, so with gender, let's talk about VOCs. Gender could play an important role in dictating how much VOCs we're exposed to, let's say through makeup or cosmetic use, right? And then you add sex to that. Sex is a biological variable, so it will dictate, for instance, skin thickness. So how much of this chemical that we're exposed to could permeate through the skin or through inhalation. And so both sex and gender could eventually affect the health outcome of this exposure. And so some of the factors that influence sex differences in research basically originate from this genetic standpoint that males have an XY versus females have an XX chromosome, and that eventually lead to different phenotypical characteristics, as well as different hormone levels, including estrogens versus androgens, as well as reproductive elements, including pregnancy, lactation, and hormone replacement therapy, etc. So there have been some studies that have looked at sex differences with pop exposure in epi studies. So, but these, for the most part, they're lacking because a lot of the epi studies or occupational studies that were done in men who were electrical or pesticide workers, and back in the day, those were predominantly men. But there were some studies that had come up over the years that have shown these evidence of sex-dependent effects with, at least with Pcb exposures, where one study showed that diabetes was 2 times more prevalent in those Yucheng women from Taiwan that were exposed to Pcbs, and that girls that had prenatal Pcb exposure tend to be more obese than boys in a children cohort study. So our group, using the CIEHS data that we have also kind of looked at these Pcb levels in males versus females. And we saw that in both the initial study as well as the follow-up study of the Anniston Community Health Survey, which is basically Anniston community residents that were exposed to high levels of PCBs and pesticides. We found that females tend to have higher levels of PCBs as well as pesticides, which kind of like laid the ground for us to look at sex differences. Other studies that we've done in our animal models to look at sex differences with Pcb exposures was this 2-week study where we exposed male and female mice to a mixture of a dioxin-like Pcb, which is 126, and the aerochlor 1260, which is a non-dioxin-like Pcbs. And so we did see an array of effects that was just, based on biological sex differences alone, and then you throw Pcb to the mix and you see even more differential effects. But what I would like you guys to focus here is to look at these red points over here. So these are the effects on the liver in the females versus the males. So it looks like in our acute study of Pcb exposure, the female mice seem to be more susceptible to Pcb-mediated inflammation and steatosis compared to the males. And this is very interesting because in the past we've done all our study in male mice, and we've seen this, exacerbated exacerbation of diet-induced steatosis and steatohepatitis, but this is all done without a second hit. So this is all done in a low-fat diet model, and the females appear to be more susceptible. So some of the mechanisms that we thought after doing the study was that, they were both endocrine disrupting chemicals, PCBs are, but they seem to be more, females seem to be more sensitive to it. And when we look at sex hormone receptor activation in these liver of these mice, we found that this PCB mixture that we were using, which was highly chlorinated, seemed to inhibit estrogen receptor expression. It seemed to have this anti-estrogenic effect and more of a pro-androgenic effect in females. So more of a masculinization effect in females, but nothing in the males. So we also did another study, and this time we switched to a different chemical, which was not a POPs, but a vinyl chloride, which is a VOC. And this is a little bit different study in that we had it as a long-term study. It was a 12-week exposure study, and we added high-fat diet to it. And so the main finding from this study is that we actually found that the females were more protective from the vinyl chloride-induced toxicity along with high-fat diet. And so the males seem to have increase in liver weight, increase in ER stress, as well as increased metabolic disruption, but we didn't really see much of these effects in the female mice at all. And our female mice actually did not seem to be very, have a fatty liver, even with a high-fat diet feeding. And I think that is something that is well known for this particular strain of mice, which is the C57 Black 6 mice. And it's thought that one of the mechanisms that these female mice are resistant to this lipid accumulation with high-fat feeding is probably through the role of circulating estrogens and how protective estrogen is in terms of combating oxidative stress and also facilitating lipid breakdown. And so we thought about this a little bit, and we thought, okay, maybe there are differences in BC metabolism in these animals that could lead to these sex-dependent effects, as well as, inhalation of breeding frequency, because these mice were exposed to VC through inhalation. But I also started thinking, if we're seeing these drastic sex different dependent effects, what if VC also acts as an endocrine disruptor, you know? And there's not really much in literature, showing how if VC or other VOCs could impact the endocrine system much. But we went ahead and took out the mouse testes, as well as the female reproductive organs, and we just kind of weighed them. And we actually saw that vinyl chloride exposure in the male mice decreased testicular weight. And so I said, okay, now I'm going to start looking at the androgen and estrogen receptors, etc. So female mice did have increase in estrogen receptor alpha expression, but not the males. And I believe there was no effect in androgen receptor expression. But somehow this kind of pointed out at this, this likelihood that VC could also be an endocrine disrupting chemical. We also did a RNA-seq on these liver samples from these animals. And similar to what we saw phenotypically, you could see here, this is just a table of all the genes that were changed or up and down regulated with each group. So I just want to draw your attention to this particular group here, comparison group here, which is the males that were on high-fat diet at vinyl chloride exposure versus the male on high-fat diet alone. And there were about 715 genes that were changed. And if you compare that to the females that were on high-fat diet with vinyl chloride versus high-fat diet alone, there was only 39. So that was a huge and massive difference in these numbers. And so clearly the males seem to be more susceptible. So other mechanism that we're thinking now that could play a role based on these findings from our RNA-seq is that perhaps there is this immunomodulation in terms of sex differences with vinyl chloride exposure, and perhaps the androgen receptor signaling has been compromised. And this is important because other studies, immunological studies that have looked at the role of sex hormones and sex hormone receptors have shown that, because I'm so much more focused on females and women's health, I forget about what is going on with the androgen and testosterone. So there's been a number of studies that have shown that males tend to have less robust of an immune response, basically because androgens could help promote the Treg population through FOXP3 expression. And so that is something that we're currently looking in the study to kind of evaluate if these immunomodulation are real or not, or if there's any basis for looking at them. So switching gears again to our EPI study. So I had talk a little bit about how in the HEAL study, we found that there was these correlation between VOC metabolites and liver disease biomarkers. So we never really completed that study that we had published. We did look at, sex differences in terms of liver disease biomarkers and VOC metabolite levels. And we did see that at least in liver disease biomarkers, the females had lower levels of liver enzymes. And we had thought that, okay, probably they were protective. So we went ahead and completed, looking at these sex differences analysis. And what was really interesting about the study is that we actually found that in females, there were positive associations between these VOC metabolites, again, with that liver enzyme called ALP. And that was absolutely lacking when we look at our male population. So it actually, kind of made us rethink that we've had all this time thought that with VC or VOC exposure, females are more protective, but we're actually seeing the opposite. And so we were interested in knowing why that was happening. And so we picked a couple of these VOC metabolites that, after adjusting for interaction between VOC levels and sex, we found two metabolites that still showed positive association with this enzyme called ALP. So one is the acrylamide metabolite AMA, and the other one is the proton aldehyde metabolite. Kind of did these dose response analysis and found that, so if you're looking at low doses, there wasn't really much of a change in the females, but they seem to be more responsive at higher doses. So I think this is the biggest finding from our study, and I think it's very impactful because, we've been talking about these residential low doses, and we've looked at our animal models with low doses, but actually, if you're talking about increasing exposures, etc., the females seem to be more susceptible. And in fact, there is a study that was done, I don't know, maybe 4 years ago that had looked at this ALP levels in nail polish workers. So these are primarily women that work at the nail salons, and they actually showed evidence that, you know, these participants of the study actually had increased liver disease biomarkers as well. And so this study kind of makes us think about some of these chemical exposures and how they could impact certain disease endpoints that could be, you know, specific only to one sex or one gender. Some other emerging new concepts. We're almost done. So one of the other new emerging concepts is altered microbiome. So again, this is a more recent exploration of what I have. I work a lot in liver and kind of branch out looking at other organ systems. And so we were interested in looking if Pcb exposure at all, long-term Pcb exposure at all, could change gut bacterial composition. And so we picked a study where we had normal wild-type mice, as well as CAR and PXR knockout mice. So these are CAR and PXR receptors that Pcbs are known to activate. So we had initially designed a study thinking that if we have these knockout mice and Pcbs cannot activate them, perhaps it would be more protective. But that was not the case because, as you know, whoever works with knockout models know that it's more complicated than that. But we did look at gut microbiome composition in this particular study and saw that there were these massive alterations in gut microbiome composition in both in knockouts as well as to a lesser extent with Pcb exposure. And this led us to also sort of, you know, move forward looking at if there is any disruption in the gut-liver axis and if that could potentially be a mechanism for Pcb-mediated fatty liver disease. And this is really, the gut-liver axis disruption is also interesting because it is now thought to be one of the primary drivers of this new terminology of fatty liver disease that has been developed in the last few years, known as metabolic dysfunction associated liver disease, or MAFLD. And I think our chemicals that we're talking about, these metabolism disrupting chemicals, to an extent also induce, could also, you know, be categorized as chemicals that promote this MAFLD. And so our preliminary studies looking at sex differences in gut microbiome. So this is again the 2-week Pcb exposure study where we look at fecal microbiome composition, and we found that in that study, so phenotypically the females seem to be more susceptible to Pcb-induced hepatic toxicity, but they also seem to have the lowest alpha diversity, which is diversity within the particular group compared to any of the other groups. And if you look at beta diversity, which is bacterial diversity, with regards to every other group, there were very distinct, there were very distinct compositions. So all of them were different from one another. And just looking without exposure, just looking at the males and females, there are distinct microbial profiles as well. And so we did look at some of the gut permeability markers in this particular study, and also found that the females seem to be more, to have reduced levels, a decreased level of some of the genes that are associated or that could encode for proteins related to gut barrier function. How did we look at the hepatic proteome, just looking at liver proteins to kind of understand, you know, how the liver and the gut work together? We did a hepatic proteomics analysis, and again, the take-home message from that was that based on sex, there were very different protein profile in the liver, and also with Pcb exposure, there were also distinct protein profile. And one of the things that I want to point out here in this presentation was that we did a downstream pathway analysis based on these upregulated or downregulated proteins. It's known as the interaction by protein function. And one of the things that really jumped out to me when I was looking at this data was that one of the Pcb receptors, the AHR, it had this activation level that was very different in the males versus the females. So the females seem to have a more robust or higher AHR activation than the males. And when we were looking at a protein list as well and seeing these upregulated versus downregulated proteins, we saw that a number of AHR targets were really high in the females, irrespective of exposure. And one of these targets was this enzyme called as flavin monoxygenase, or FMO, FMO3 in particular. And you can see here, there was this distinct sex differences where in females, FMO3 was expressed about 3,000-fold higher than in the males. And this is also validated with Western blot analysis of protein expression. And so based on these findings, we are now looking at another arm of how this gut-liver axis story might pan out, at least in terms of PCB exposure. So FMO3 is one of the enzymes that help metabolize this food precursor known as trimethylamine. That's usually the body usually make from a carnitine-rich food such as eggs and fish. And so it gets metabolized into this chemical trimethylamine oxide, or TMAO. And TMAO is a very well-known biomarker for a number of cardiovascular disorders. But more recently, people have also shown that TMAO might induce ER stress in the liver and promote metabolic dysfunction-associated fatty liver disease. And so that is one of the mechanisms that we think might be playing a role driving these sex differences with PCB exposure. And so based on all those findings, looking at the gut microbiome, endocrine disruption, there's this evolving concept that I've been working on known as the endocrine gut-liver axis, and how exposure to chemicals could alter all these three systems together and trigger this tri-directional relationship leading to metabolic dysfunction. And this is brought upon, easily because of these receptor-receptor interactions, the role of sex hormones in mediating energy metabolism, liver function, as well as gut microbiome composition. And so once I figure out how this works, hopefully I'll be back for another seminar to talk on that. But I also want to mention about other ongoing studies that we're doing. So we've talked a lot about PCBs and vinyl chloride and VOCs. So more recently, we've been working with another organic chlorine pesticide called chlordane. So what's distinct about it is actually it's a mixture of different chemicals. And this just, brings me to start working more with mixture of chemicals, because we are never exposed to just one compound or one chemical alone, but to an array of chemicals. Other things that we're working on is also to develop these multi-omics approaches. So you've seen me, present on transcriptomics, metagenomics, as well as proteomics, and how to integrate all that information together to get a more holistic approach on what's going on in terms of disease endpoints. And another thing that we also are planning on doing, or I'm planning on doing, is also to start using mixtures that are more relevant to today's exposure patterns. So looking back at our NHANES database, our HEAL studies, as well as our CIEHS studies, and trying to identify what some of those chemicals are higher in composition or higher in circulating levels, and develop that mixture to look at our animal models. I'm just going to show you this one slide where we're looking at organic chlorine pesticide exposures. So when I was talking about how when we're doing sex differences, somebody was saying that we're not doing it right. And one of the right ways to do it is actually when you're working with female mice is also to try to monitor their estrous cycle. And the reason for that is because, at least in our case, we are working with endocrine disrupting chemicals, and they could eventually disrupt estrous cycle in female mice. And so this was the design of the study. It was a low-fat diet study with chlorine exposure for 2 weeks. And so a normal estrous cycle in mice and rats usually is about 4 to 5 days. It's broken down into 4 phases: the proestrus, the estrus, which sort of resemble the ovulation phase in humans, the metaestrus and the diestrus. And based on the types of cells that you see using a vaginal smearing on these animals, you could identify which kind of estrous cycle it is. And I was wanting to show you this data, but unfortunately the colors are not working on the screen. But what I wanted to show is that with chlorine exposure, what we saw with these preliminary studies was that it kind of pushed these female mice to go in a longer estrous cycle compared to our control mice. And we also saw evidence of both metabolic disruption and endocrine disruption with this particular pesticide exposure. So in its conclusion, we covered a broad range of mechanisms, And I had talked earlier about a two-hit hypothesis where I thought that originally we needed a alcohol consumption or a high-fat diet and then a chemical exposure as a second hit to induce these hepatic endpoints. But after looking at our mechanisms, we have been redrawing on the whiteboard and thinking that it's actually the other way around. So these chemicals actually look like they are the ones that are causing this first hit. So we like to call it a compromised liver, so you're growing up, you're exposed to these chemicals in utero during childhood and adolescence, and all these things are happening, these, subtle changes, metabolic disruption, signaling disruption, gut microbiome alterations, leading to a compromised liver. And then as we keep growing in life and we're exposed to lifestyle and social factors like nutritional stress, alcohol consumption and smoking, etc., those could act as second hits or multi-hits, if you will, to drive metabolic dysfunction and toxic and associated fatty liver disease. And eventually, this could result in multi-organ toxicity as well. And so with that, I just want to talk a little bit about my research interests. I'm an early stage investigator in the field of environmental metabolic health. I'm very interested in sex differences as well as environmental women's health. And hope to focus more on health minorities and indigenous communities on a global scale. I'm also very interested in reproductive health, and I think it's even more important today in how chemical exposures could impact reproductive health, given our more recent developments where women's reproductive health have been compromised. I'm also interested in health disparities, looking at other diseases as well, cardiometabolic, as well as cancer endpoints. And also, another of my research interests is to be engaged with the greater community. So working on intervention and prevention strategies and how nutrition and healthy lifestyle could help improve health and reduce toxic exposures. And with that, I'd like to acknowledge a number of people that have helped me along the way as a scientist. I'd like to give a shout out to my KO1 mentorship team, my former mentors and current mentors, my collaborators, especially Sheshra, who has helped me with a number of analysis. And I also wanted to give a shout out to my students who teach me how to be a better scientist and a better mentor. And also the environmental liver disease group upstairs on the 5th floor that constitute of the Matt Cave lab, the Jocelyn lab, the Watson lab, and Carrie Kling's lab. Also the CIEHS for always guiding me with my career development and all the financial support with my research. And thank you for everyone for listening. And I would love to answer some questions. That's fine.
Problems in this room, so
Oh.
everyone talks loudly, and if you would just repeat
Oh,
the
yeah.
question.
Great. I have a question about your epidemiological study at the association. And so you are presumably looking at urinary VOCs as a surrogate for exposure, right? But do you really know if people with high urinary VOCs at higher exposure are they just better at getting rid of it, in which case their body load would be low and your relationship would be worse?
Okay, so Mike was asking if I know, because we were measuring urinary VOC metabolite levels, if that is actually correlate to actual VOC exposures or the parent compound, right?
Yes.
Okay, so that's a great question, and that's a question that one of the reviewers have raised also. So the answer is no. I mean, that's a tricky thing about, measuring urinary VOC metabolite levels. You do not really, it's kind of like, not really a guessing game, but kind of like a speculation that, okay, these are the sources, it's coming from these sources. So another way that we are trying to handle that is to get as much more information from our demographic data, looking at everyday exposures, occupation, just to see if really those VOCs and lifestyle factors like smoking, just to see if those urinary VOC metabolites that we're measuring really are from, let's say, smoking or, cooking or cosmetic use or something like that.
Right, my point is, the urinary VOCs could also be high too, just because these people are better at it's pretty.
That's true, yes. So there is a study that was done last year, I think, that got published last year, where they actually look at benzene metabolite levels and the rate of benzene metabolism. So it was actually shown that females were slower metabolizers than males, so they had lower metabolite levels than males. So that is a very good point of view. So we have not looked at, rates of metabolism or anything with any of these VOCs.
I mean, can you do something that we're not, for example?
I think they are developing those methods. I'm not the right person to answer that question in terms of method development, but I will find it out and let you know.
And Rita, perhaps you could mention about the NHANES study that you did with VOCs.
Oh, looking at plasma.
Plasma levels.
Oh,
The answer is
all right. So so I'm not sure what method they use, but in our current NHANES study where we are actually looking at plasma VOC levels, unfortunately, we're not seeing a lot of detection rate in a huge number of the population. So, which actually is putting our study back. So it looks like so far with what we have, urinary VOC metabolite levels are still better. So, but hopefully in the future that might change. Okay.
It's not online.
I do have one more question. So you did some proteomic studies and some transcriptomic studies. How do you see the two line up? Are there a lot of similar transcripts and proteins going out for down in the same direction?
Okay. So in this talk, whatever I showed were from different studies. So transcriptomics was from a different study, and proteomics was from a different study. But Carrie, our work with Carrie, we've done looking at the proteomics, transcriptomics, as well as microomics, if you will, eventually. And so what we did with that, so that is a multi-omics approach to where you're doing different omics in the same organ system, right? And so they have, I think we did integrated analysis. So looking, I don't remember what Shesh did exactly, but I believe there was some integrated analysis looking at if this gene expression correlated with this protein expression. So, and we're doing that the same thing with microRNAs and mRNAs as well.
Thank you very much.